how many.
Right. Okay, so we're ready to go.
Okay, so let me get my slides, share the screen again. So in this last talk, I will talk about breaker. And first, let's say, what is breaker? So breaker is a gene annotation pipeline, which is combining GeneMark with another annotation tool, Augustus. And you may ask why we want to use something else than GeneMark, since GeneMark is giving us such good results. So firstly, developed breaker 1, combined breaker ET, which is using rna-seq data with Augustus, and it combines the strength of both of these algorithms. So one of the strengths of Augustus, which GeneMark doesn't have, is its ability to predict alternative isoforms. So while GeneMark only predicts one protein isoform per gene, and hopefully the main one, Augustus can predict multiple ones if they are supported in the RNA-seq data. Another difference is that GeneMark is using RNA-seq information in training, but it's not really using it directly in the predictions. While Augustus, if it sees a strongly expressed intron, it will try to always predict it, no matter how the model estimated the gene structure before. So it's kind of directly using the external RNA-seq data in predictions. But the main weakness of Augustus is that it belongs to the family of supervised trainings algorithms. So it depends on manually created gene sets. However, by using GeneMark ET, which is self-training, you can overcome this problem by using the GeneMark ET result in substitute of the manually curated gene set. So by combining these two tools, you get an automatic gene prediction tool, which combines the self-training strengths of GeneMark and the strengths of Augustus, which consist of alternative isoforms, utilizing data in predictions, and so on. So as you can see, this is another quite popular tool used by multiple groups to annotate eukaryotic genomes. And just to give you a better idea of the overview of what breaker does. So it takes your genome, it predicts genes with GeneMark, uses it to train Augustus, and predicts genes with Augustus. It's really that simple. So this was breaker 1. Second is a breaker 2, which is doing the same thing, but now it's using proteins. So it's connecting GeneMark EP+, which has been introduced, which is able to utilize proteins from even remote homologs, and it's combining with the Augustus, which has, again, the same strengths as I already talked about. And what makes breaker 2 different from other tools which are using protein information is mainly its ability to utilize the remote homologs. So many algorithms are able to take a very closely related protein and infer the gene structure from it. But not that many, or at least no algorithms that we know of, are able to take proteins which are more distant than maybe the same taxonomical genus and still extract meaningful information from these proteins and use them to improve training and predictions. And the reason breaker can do it, it's because it's using ProtHint, again, for protein mapping. And ProtHint is treating the proteins not really on the level of global, of protein alignment of the whole protein to the gene, but it's treating it on the level of individual introns, starts and stops. So whenever there is a conserved domain in a very otherwise remote protein, ProtHint will still be able to catch this information and use maybe 1 or 2 introns out of a otherwise very long gene to help improve the predictions. And this can be used to improve both predictions and training. So this is one of the reasons why breaker 2 is a useful tool. And to introduce breaker 2 in more detail is to show you this diagram, which may look scary at the first glance, but it's not scary anymore when you realize the whole section here is again showing ProtHint, which was already introduced in the last presentation. So you want to again focus on the selection of training genes for Augustus and then predicting genes with Augustus in this case in iterations, which makes breaker 2 different from breaker 1. And it's done in iterations because if you remember, we used to initialize prod hint with gene predictions from GeneMark ES. And I mentioned these predictions can be noisy, but it's fine as an approximation. But in this case, if we use the output of Augustus, which is output of the first big iteration of breaker, to initialize prod hint again, you can get slightly better estimations, not slightly actually, much better estimations of the gene regions, and then run the whole pipeline again. And in second iteration, get a better result. So this idea is again a bit similar to the self-training concept introduced all the way back in GeneMark ES. And I also want to highlight runtime. So our goal here was to develop a tool which is executable on personal machines. So you can see that even on a quite large genome of tomato, Solanum lycopersicum, which is over 1 gigabases long, and when you use all plant proteins, which in this case in OrthoDB is 3.5 million, you can still run this under a day on a laptop, on a machine with 8 CPUs and 8 gigs of RAM. So that's another big strength of Breaker, that you can quickly analyze a large number of genomes without powerful hardware. And I want to stop. Oh, someone's leaving, so goodbye, Pedro. I want to quickly stop and explain the procedure for selecting training genes. So it's explaining what's going on here with training gene selections. So I said that Augustus is using the result of GeneMark EP or GeneMark ET to train, but that's not 100% correct. It's doing some filtering to make sure it's only training on genes which are actually correctly predicted. So again, we can take a look at this graph on this diagram. And on this track, we see GeneMark EP plus predictions. And here we have the total of 6 predicted genes. However, not all of the predicted genes find good support in protein data. For example, this gene is not supported by proteins at all. We were able to map a start codon, which is symbolized here, but the mapped start codon is not even matching the predicted start codon. So this gene is suspicious, to say at least, so we do not want to use it to train Augustus. This gene, on the other hand, has matching start codon in protein data. It has a matching intron. All of the introns are matching perfectly, and it has a matching stop codon. So we are much more confident that this gene will be correct, and we want to use this gene in training. So out of 6 genes here, we have some external support for 3 of them. So we want to use 3 genes here for training of Augustus, and thus ensure that our training set is really confident, without noise, and reliable. And this diagram also illustrates the second point I mentioned about alternative isoforms. Here you can see that Augustus for this particular gene was able to predict 2 alternative isoforms when it saw evidence for some exon skipping in this gene. So one of the alternative isoforms completely skips this exon. And GeneMark EP is not able to predict this. Augustus can predict this without any issues. So that's one of the reasons why they are combined together. And to put more emphasis on the improvements along the steps of Breaker 2, I want to show you kind of how the algorithm develops from beginning to the end. And there's a lot of numbers. Let's just focus on the harmonic mean of sensitivity and specificity on the gene level. So this is an example for Drosophila melanogaster, and we actually excluded the proteins of Drosophila melanogaster itself from the database to make the run fair, because we don't want to be utilizing the proteins which we are trying to predict to help us. And we can see that starting with GeneMark ES, which is using just the sequence alone, we can improve by about 15 percentage points by the usage of GeneMark EP+. Then when we use GeneMark EP+ to train Augustus and predict genes with Augustus, we can jump additional almost 10 percentage points to arrive at a better prediction. And then we can have a slight increase when we do yet another iteration of breaker. In this case, the difference in iterations was pretty small. And it's also a reason why we only iterate once. So if we saw a bigger improvement, it would make sense to iterate 3 or 4 times, which can be done. You have the option to iterate as many times as you'd like, but we think that to also balance the runtime and accuracy trade-off, just one extra iteration is enough. And now in the final slide in this short presentation, I want to compare breaker 1, which is using RNA-seq data, with breaker 2, which is using protein data. And the comparison is really dependent on how close the proteins on your input are. So in some genomes, even with remote proteins, you can get better results with breaker 2 using only proteins than with breaker 1, which is using native RNA-seq. So this result was kind of surprising to us, and it may imply that the organization of genes in plants is more similar than organization of genes, let's say in insects, that proteins here from a different taxonomical order were still enough to help us more than the data from RNA-seq. One of the reasons also which this might be the case is that from RNA-seq, you only get locations of introns. I forgot to highlight that from proteins, you also get the N and C terminus of the protein, so you can directly map where the protein coding region starts and where it ends, which is information you do not get from the transcript data, because the transcription start and translation start are 2 different things. So from RNA-seq, you get a rough idea about transcription start. From proteins, you actually get exact idea about translation start. So that's one of the reasons why using proteins is sometimes better, but not always. As you can see, in the case of worm, C. elegans, using proteins is only better when you have availability of proteins from the same genus. Once you don't have proteins from the same family or order, you are actually better off with using RNA-seq. So obviously what you want to do if you have both sources of information is to use both and hopefully arrive at some point which would be here, something which combines the strengths of both of these approaches. And this is a project which we call Breaker 3 now, and that's work in progress to have a good way of integrating both RNA-seq and protein data to improve the prediction accuracy. So these are the slides, and now another demo, but as always, please ask some questions before I start. I think no questions, so let's see what the tutorial on breaker has. So it's a 3rd tutorial on this website, introduces you how to annotate genes with breaker. And here we will use the fact that we already have annotated Arabidopsis with GeneMark EP+, and now we will try to improve the annotation by using breaker on the same genome. And to illustrate also another point, I talked about RNA-seq a lot, but I actually never showed you how to use RNA-seq, so I will, in this case, use both RNA-seq and protein inputs to show you how to use them in breaker. So to get RNA-seq data, first you need to get some RNA-seq reads library. So normally you would use your own. Probably you did RNA-seq experiment together with your sequencing experiment, so then the RNA-seq reads would be something you internally have. For this example, because we are working with publicly available data, pretending they are our own, I chose a random RNA-seq library from NCBI. You can see the details here, and downloaded this RNA-seq library with SRA toolkit. So this can be done just with 2 commands. And after you run these 2 commands, what you will see are 2 files. And these 2 files will be read, paired, and reads in the FASTQ format. And these needs to be aligned on the genome to give you an idea about introns and transcripts and expression and so on. So in this example, I'm using STAR, which is one of the most popular RNA-seq reads aligners, but there are many other alternatives. HiSet2, which is fast. I think it's faster, less resource intensive, but less accurate. And because we have time, so I decided to use STAR and map the reads to the genome more accurately. But I have to warn you that STAR can really consume a lot of resources, especially a lot of RAM. So you can only use STAR when you have a lot of memory available. With that said, STAR runs in just 2 steps. You first need to prepare a database from your assembly, and then you just run STAR itself. And there are a couple of options which are worth highlighting. One is that STAR needs you to specify minimum intron and maximum intron length to align. So these values, 20 to 500,000, are kind of default values which should suit most of the eukaryotes. However, if you know that your genome will have much shorter introns, or on the other hand, you expect that the minimum intron length would be much longer, you may want to adjust these parameters. And we are also running STAR in a 2-pass mode, which is important to mention that the usual run of STAR expects you to supply a gene annotation. And then it uses this gene annotation to improve mapping. However, because we are interested in creating a gene annotation here, we need to run it in a 2-pass mode, which basically in the first pass, it approximates the locations of transcript from the RNA-seq data. And in the second pass, it will use this internal gene annotation, which would be very rough, but still better than nothing, to actually align the reads. So maybe this is the most important part from this short section, that you want to use 2-pass mode in STAR to discover novel splice junctions. Then the gene prediction with breaker itself is again designed to be very simple. So breaker actually runs with just a single command, and you need to specify your genome. Here, you want to specify the hints, which we already have protein hints, so you can reuse the protein hints from prod_hint, which we generated in the other example. However, let's assume you only have protein sequences, you want to use them directly, so you would replace this hints parameter with protein sequences parameter. You can also specify RNA-seq data and say you want to run in this ETP mode, which is still an experimental mode which tries to combine results from protein and RNA-seq mapping in both GeneMark and breaker, but right now it's pretty much only looking at introns which are found in the intersection of both protein and RNA-seq data, and it weights such introns with much higher weight than other introns because they are found in 2 independent sources of evidence. So this is kind of the first attempt to use RNA-seq and proteins directly together, but there will be more improvements coming. So once you run breaker, it will run for about, in this example of Ataliana, for about 8 hours. And the runtime would be slower if you actually run it with protein sequences from scratch, because this option --hints enables it to reuse the protein hints, so you can skip the whole protein mapping steps and directly use it. So you see, you can also specify the result of GeneMark directly in breaker. So you can specify Augustus model directly in breaker. So all these components are very flexible and enable you to either automate the task as much as possible by just running a single command, or you can also run most of the tools outside of Breaker, but then kind of use Breaker to finalize the annotation, use everything together, and get the final result, which gives you more flexibility, but you need to know what you are doing for that. And with that, you will get a Breaker result. And you can either, again, evaluate it against a reference annotation or another gene prediction set if you have one. If you don't have one, you can again use Busco in exactly the same way as we used before. And here we want to use, okay, what changed between the results of GeneMark EP and Breaker, right? So GeneMark EP already did pretty good. And let's see how it did compared to Breaker, if there was any improvements. So we will show on the left the results of GeneMark EP+, Busco score. Sorry for this. Busco score here was, we found 4,534 of the expected genes. And with Breaker, you see, okay, we improved. We found 4,567 of the expected genes. So there was a slight improvement in the Busco genes found, and also improvement in the overall gene prediction accuracy, as was shown in the slides. So this is how you can evaluate the result. And unfortunately, Breaker does not yet have an option to show the same kind of annotation report as in GeneMark. To show you what I'm talking about, I'm talking about this kind of report. So this is, for now, GeneMark specific, but we are working on adding the same kind of report to be specific for the Breaker output, to include maybe information about Augustus models, as well as the GeneMark models and some additional information, but that's work in progress. What's also work in progress is the selection of a reliable gene set. So this is not automated in Breaker, but in case you still want to do it, or you still want to select genes which are supported by external evidence, I show a procedure to do this step by step, kind of manually. And this is maybe good to show because it introduces more concepts about the output formats and comparison of the results. So I will just go through this step by step to show how to do this task manually, pretty much. So when you want to select a subset of genes supported by external evidence, you may compare the result of Breaker, which is called Augustus with hints in this case, against the hints you collected during the Breaker run. And you want to specify you only want to get introns, so you only want to compare introns, and then you specify where you want to output introns which are matching between both of these sets. So this gives you a file with introns which are found in both the Breaker prediction and they are found in the external evidence. And from these introns, you can extract gene IDs of genes in which these introns were found. So now you have gene IDs of genes which had introns in the external evidence. So in other words, you have IDs of genes which are supported by introns. You can do the same for starts and stops. And then all you need to do is grab these gene IDs from the Breaker result and get a supported subset. Again, what you will see is that in your supported subset, you increased specificity. Here you pretty much removed almost half of the false positives predictions at the cost of losing about 1,000 of correct predictions. And similar trend can be observed on the exon level. There is a script coming which will do the same for fully supported genes and will automate this process, but that's again work in progress. And now we have 4 minutes until the end, so I think that's good time for questions. I won't start any new topic now. The only topic left are common issues with Breaker, which are well documented on Breaker's webpage, so I will ask you to kind of look at them on your own. And just to show you I'm not lying, common issues of Breaker are documented here. And some of them are the exact same issues shown for GeneMark because Breaker runs GeneMark, so solution to these problems would be the same. Some of them are more related to connection of GeneMark and Augustus. The main problem we have with Breaker is that whenever either GeneMark or Augustus is updated, it may be incompatible with the other two. So Breaker usually adapts very quickly, but users who installed a version of Breaker and then they may be updated Augustus or GeneMark, but not the Breaker itself, then the program would not be running for them. So what I would recommend when you encounter a Breaker issue would be to first try to update both GeneMark and Augustus and Breaker, and then usually the problem is already fixed in the latest release. So this would be the most common issue you can encounter with Breaker. So this is all, and now I would be happy to hear the questions from you.